our webinar.
Great. So thanks, John, for making the introduction. So my name is Mark Atkinson, and I am head of the Diabetes Institute at the University of Florida. And together with John and some other investigators we'll introduce in a minute, I'm one of the, again, co-principal investigators of the CaRe T1D program. So today, the goals of today's call are listed here, but we wanted to provide everybody a background of the CaRe T1D program, where it is to date, and where the thoughts are in its future, and then also to help you. And you'll hear more in a moment from NIH about the RFA that's coming out. But if you're interested in applying for the program, we wanted to provide you with background information in terms of what types of cases are collected, how many cases will be available, the tissues available, how they're processed, database information, which John has been taking the lead on. And then again, we'll close the call today with an NIH funding opportunity, more information on that, and how you'll find out about that. And if we go to the next slide, I'm going to introduce somebody that I've worked with for almost 3 decades now. I've hit my 40th year in type 1 diabetes research, and I've had the pleasure and honor of working with Marjorie Perry. So Marjorie?
Thank you, Mark. So I have a daughter with type 1 diabetes who is unfortunately at the age of 41 developed some very serious cardiovascular disease that was completely undiagnosed, and it could have been disastrous, but thankfully it wasn't. But I'm thrilled about this initiative because cardiovascular disease is the leading cause of death and morbidity, in type 1 diabetes. And there aren't any treatments that exist for this. And clearly, the animal models are very important, but we definitely need to study human tissues for human diseases. And that's why this is so important. And there are very big differences between cardiovascular disease in type 1 diabetes, people with type 1 diabetes and people with type 2 diabetes, and it's just not very well defined at all. And doctors actually are not attuned into the needs of the type 1 diabetes community. So this program really will use these human tissues that are so important to find new treatments and really promote the outcomes that we definitely want for cardiovascular disease and type 1 diabetes. So I'm thrilled to be able to do this. And thank you, Mark.
And thank you, Marjorie, for setting this up. So now we know why we're here. And I want to thank everybody on this call for joining. A great turnout. Next slide, please, John. So the NIH recognized this problem a few years ago and set a priority on this. And I want to make sure it's clear. It wasn't just NIDDK. It was NIDDK and NHLBI that came up with this idea that we need to make progress in this area for the reasons that Marjorie just said. And so back in 2021, they advertised this RFA for applications that would have a due date of October in 2021. And it was for a cardiovascular biorepository set up for type 1 diabetes. And this would be phase 1 of what would eventually be a 2-phase process. And you're going to hear in a few moments, and that's the opportunity for those on this call to participate in the phase 2 operations, which is studying these tissues. Next slide. Please, John. All right, so this is the leadership of the group here, and you've already met or heard from a few of them, but I want to again acknowledge our, I'll say, CaRe T1D family here on the left-hand side. This group has been meeting routinely and often ever since the initiating of the program, and the NIH officers are listed on here. Most of them are on the call today, and you'll have a chance to speak with them later on. The biorepository has 4 sites associated with it: the University of Florida, the University of Alabama at Birmingham, or UAB, Vanderbilt University, and then UPenn with, again, John taking the lead here. The expertise fell along the lines of experience in terms of collecting organ donors and tissues and processing them. With UAB had expertise in cardiovascular analysis. Agnes Fogo and her team at Vanderbilt University in terms of the kidney. And so it was really, I'm honored to work with all of these people. And I'll also mention Des Schatz, who's our pediatric endocrinologist and type 1 diabetes expert. But we also, as part of the program, sought to have a expert scientific advisory board. And a lot of what you're seeing, you will see in the next few moments. And my presentation will be brief, I promise. Benefited much from the advice of these individuals. Next slide, please. So what was the goals of the biorepository? And many people here have probably put in an NIH grant. I've, as part of the prepping for this meeting, I cut these words down, but the bottom line is pretty obvious, that to establish a state-of-the-art cardiovascular biorepository for specimens from organ donors with type 1 and type 2 diabetes, as well as organ donors without the disease, that would be age matched and as well as possible. And then as part of the biorepository functions, you have the recovery, the processing of tissues, the case assessment to make sure that they are what we think they are in terms of the disease form, and then assuring quality QA/QC for all the samples. And then the second aim is to distribute those, and this is where those on this call will come in, is to approved investigators, and then have data, and John is working hard on this, into a multimodal database, and then to promote collaboration amongst what eventually will be this CaRe T1D community. And if I didn't mention it before, it was on that first slide. CaRe stands for Cardiovascular Repository in T1D. But again, the emphasis here, as Marjorie said, is to better understand differences, potential differences between cardiovascular disease and type 1 and type 2 diabetes, with the hope of improving patient outcomes. And again, as part, that's why these bottom functions will, aims, two functions will become important, including one of the things that we'll do is to organize an annual investigator meeting where the data and discoveries will be presented. Next slide, please, John. Now, how does this operate? And again, I know Sanjoy Duda from JDRF is on the line. 17 years ago, an organization, if you're not familiar with it, called JDRF, or Juvenile Diabetes Research Foundation, took a risk and started a program called NPOD. And the goal there was to recover pancreases and other tissues of interest from organ donors with type 1 diabetes. And I could spend an hour on this slide. I'm going to try and get through it in 30 seconds. But it's a group that operates 24/7, 365. There's 56 organ procurement organizations in the United States, and we work with every one of them. At any given time, we'll have up to 17 people on call. We work every day of the year, Thanksgiving, Christmas holiday, whatever your holiday is, we're on it, even through hurricanes. And the goal here is to identify organ donors that meet acceptance criteria. We started out with the one program on pancreas, but now we have 6 programs that we're involved in. And again, there's about 13,000, a little over 13,000 organ donors every year in the United States. And we, depending on the year and our criteria, we receive donor offers from anywhere from 600 to 750 of them. So we receive many organ donor offers, and we accept them based on criteria that I'll show in a minute. But again, oftentimes people will refer to NPOD as being autopsy. These are not autopsy tissues. These come in as if they're going for transplant. So very high quality. And again, we're very, they come in with families consenting these for research. So we're very blessed by that. Next slide, please, John. And here's the criteria. And we had some initial ones that were put in the grant application for the biorepository, but after consultation with our scientific advisory board, they were modified. And these will be up, and John's going to post them. So if they help you in terms of your application and what you think might be available, this is the group that will be there. And I'm going to mention this in a minute. The goal for the biorepository was within the first 2 years to have 60 total cases, 20 type 1 diabetes, 20 type 2 diabetes, and then 20 control donors. We're on target. We're at almost 40 cases now. And the goal is, again, by October next year to have 60. So we're moving in on that. But these are the criteria. And again, these will be posted and you can see them. Next slide. And here's how the program works. Again, what happens is we're contacted by these organ procurement organizations or OPOs, and they have an option. Some of these cases, depending on the tissue, they will go to transplant. And in other cases, for a variety of reasons, will come to us for research. And what our goal is that, again, you will, on the investigators part, will apply, and we'll have a simple application process that will be reviewed internally by a scientific advisory board on a regular basis. There's usually a very fast turnaround here, and as long as you provide, evidence of IRB and a limited preliminary data that you have a scientifically valid idea, it'll go for project approval. And then what you'll have is access to this biorepository where we'll have blood, kidney, heart, and peripheral vascular tissues available for you to do your analyses on. The clinical data from all of these cases, and there will be much of it, will all be stored internally, meaning the core lab has a series of functions that we'll show you in a moment. But then these are the types of samples that'll be available, from FFPE blocks, fresh, frozen, samples for EM, and then even the potential moving forward from some fresh tissue that we'll talk about. And we do some micro CT analysis to look at calcification. The goal is here, again, to generate publications and data, not just publications, but data that will be transformative to the care of cardiovascular disease and diabetes. And again, John's going to present his data in a moment. Next slide. So how are tissues processed? And again, we're thankful Dr. Kusmart-Siva, who is on the Zoom here today, and she and her team do all of this processes. And again, this information will all be on a website for you to look at. But again, what we do, we collect whole hearts and whole kidneys. And again, they're anatomically designed so that we can tell you what region they were, not just the case, but what region the tissues derived from. So if you are looking at a location-specific event, you'll be able to request tissues from this. And again, from all of these samples that are collected, a series of analyses are performed. They'll be web-based, so you can analyze yourself and say, I want this case, I want that case, etc. Next slide, please. So what am I talking about? So this is an example of a heart collection and how they're processed in terms of the coronary artery tissue collection and then the ventricle tissue collection. So again, on I don't go back, John, but you will have from these various regions within the heart, tissues processed by that variety of means, again, within these patient study groups. Next slide, please. And then this is, again, just a couple examples of core lab analysis. A series of specialty stains are performed on every case, both in the kidney and in the heart. And for every case, all of these are scanned in and you'll have access to a website where you can do your own pathological evaluation. And then again, we'll have the equivalent of like a Amazon shopping cart where you can go in and say, I need 5 slides from this block or this region or whatever. And it's all an automated process where you put your information and click a button, and then it goes to lab. You'll receive confirmation. Again, this is what happens when with 17 years of experience on this and a few bumps along the road, but we try and make it easy for end users. Next slide, please. Just a couple more. And this is another example of core lab analysis. It's not a movement here, but again, for example, on the heart, we do whole heart micro CT, do about 2,000 images in 15 minutes, and have a whole series of software that we're able to measure calcification or look at various aspects of the heart, volume, et cetera. So all of this information is and will be made available. Next slide. And a common question we get, and I'm going to turn this one over to Mingder Yang, is, well, what do I need to do to receive these tissues? Mingder, will you share that?
Sure. My name is Mingde Yang. I am a member of the administrative team at University of Florida. I'm guessing that some of you are interested in getting human samples to try to optimize your assay or generating some preliminary data to submit a grant. So we have identified a couple of cases that we can provide the samples to the investigators before you obtain the funding or submit your application to become an investigator. We do, however, we're going to need your IRB documentation as well as a simple MTA between your institute and the University of Florida. The detailed information will be available on the CaRe T1D website by January 16. Thank you.
And one more word. Thank you, Binger, for sharing that. Again, one more word here is, again, understand that because the donors are deceased, most IRBs handle this as a special form of research. And this, again, most IRBs, this is not month after month after month of application because of these are deceased individuals. We still maintain all of the privacy issues along with this in terms of the data that you will receive, but it's usually far less painful than a traditional clinical IRB. Next slide.
Okay.
Go, John, it's up to you. Thank you.
It's up to me. Okay. Well, thanks. It's my pleasure to introduce our website and database that we're actually calling a research storefront. And so the idea behind the storefront is it's a web-based application that provides investigators with information about the cases and the availability of specimens for further research. In this slide set, I'm going to provide an overview of the storefront so that you can see what the repository holds for you as a researcher with an interest in cardiovascular effects of type 1 and type 2 diabetes. So we're going to start here. These are all, by the way, these are screenshots from the website. I thought it would be most useful to do this because I've also included notes with this presentation, and these will be posted on our YouTube channel as well. So going across the top, we have a mission tab here where you can see about the purpose of the CaRe T1D program, and it lists all the key personnel that are associated with the project. And then also next, a brief statement about how we actually do our work, and a listing of the Scientific Advisory Board Committee, the Executive Committee, and also the cores. And then finally, a public service announcement about organ donation. So moving over to the next tab, this is really the key feature of the storefront, and it's the presentation of donor case data. And that's the real function here behind this explore cohort data function that you see here. So the general public can actually see a demonstration case, as you're going to find out. which is populated with synthetic data designed to be faithful to the type of data and distributions you would see once you have obtained an IRB approved protocol to use CaRe T1D data. So as you can see over here on the left-hand side, we have a set of filters that we can actually apply so that we can hone in on a particular selection or cohort criterion or a set of criteria. And then over here on the right side, you will see graphical displays of selected statistics based on the filter that was applied. As of right now, no filters are applied, but all the filters are used by just actually these slider objects that you see here. And then you can see here up at the top, you can see individual cases. Right now, we have just a demonstration case, and the reason for that is because this is anonymized data, as I've shown before. In fact, it's synthetic data. And so we're not showing any live data in this particular case. When you get the IRB protocol approved and the MTA in place, will then, needless to say, this will be populated, this whole area here will be populated with little squares that look just like this. And we can actually see within each one of these cases where we can see the details, takes us into a pretty deep dive into the data, starting with donor information, the lab results, and the high resolution HLA. Then moving to the clinical history, a description of the hospital admission course, the clinical history, medications, toxicology, a hospital laboratory panel, and then the hospital evaluation. Then next, we can see under case processing and RNA quality, and also the 3D reconstruction of the micro CT scan, which is actually a movie. And so you can play the CT movie from here. And you can also see that there's a carousel of still images from the micro CT as well. And then finally, there's a histopathology tab where you can get into the slide archives, where you can view them online using the Apereo digital pathology platform. So next, for investigators, we see there are several pages here that provide information to those who plan to use or are currently using the repository. So you can apply to join. And then you can also look at the standard operating procedures that are associated with each one of these, with each organ, actually. And then also information about the secured, information about the secured online pathology site, which is hosted at the University of Florida. In order to use the system, you'll need to set up a separate account, as noted in these instructions. And then here is the part that Mark was just talking about too, the available samples. There's quite a bit of information in this particular page, including some information about the repository itself, once again, some information about organ processing, but very much this is sort of that Amazon order type of system that we're going to be implementing here and have implemented actually, that these are samples in stock. And as you can see here, various and sundry samples identified here and then tabulated by type 1, type 2, or other diabetes or no diabetes. And these numbers are updated in real time based on the samples that have actually gone out to investigators. And then in addition to this, you've already seen these diagrams, but it's just to remind people that that's where these specimens are actually coming from, so that there's an anchor to the table that I just showed you. And then finally, there is a login for those people who have been approved for full access to all of the donor and organ data in the repository. You will have to log in with your email address and password once that account has been set up. So I do recommend that you try this out to get that live experience, and the link is right here. And I'm also going to post that into the chat so that you can copy it and examine this at will. Okay, now I'd like to turn it over to Teresa and Rahul.
Yeah, thank you. So I'm just going to briefly talk about this future funding opportunity to build a consortium. We have as Mark said, we have a biorepository that's growing and very robust, and we have this wonderful website. So now we'd like to build it out and bring the science to using these human tissues. So next slide. So we have, we're working, there was a notice of intent to publish about a funding opportunity. We think the RFA may be released this week. And so you should check the NIH guide for this. We'll also be sending out announcements about this. And we are planning, it's mentioned in the RFA, but having a webinar specific for the RFA. Well, I said mid-December, but with the delay, it'll probably be maybe the first week in January, but we'll let you know. So that will get into some of the science and the administrative aspects of the RFA, primarily to answer questions from all of you. The key parts of these applications and these U01 grants are underlined in this statement: hypothesis-driven studies on advancing the mechanistic understanding of cardiovascular disease and type 1 diabetes using the CaRe T1D biosamples and resources. So we're looking for science that's not pure discovery, pure omics, data generating research, but for projects, studies that will be driven by hypothesis. The focus should be on cardiovascular disease. We also have kidney tissues in the biorepository, but those are really to inform the cardiovascular research, because it's been known that the link between kidney disease and heart disease, but this is on the cardiovascular research. Again, we have tissues from people with type 2 diabetes or no diabetes, but the focus will be on type 1 diabetes. And of course, making comparisons, would be good. You can use animal studies to further the findings from the CaRe T1D biosamples, but we really want the focus to be on the human tissues. So it would be more like you would find something with the human tissues, and then you may want to test it in an animal studies, and then maybe even bring it back to human tissues. Next slide. So here we'd like to really build the CaRe T1D consortium. As Marjorie said in the beginning, we don't know very much about it. And so we want this to be a big step forward in understanding the pathophysiology of cardiovascular disease in people who have type 1 diabetes. So as part of this consortium building, we're going to be sharing the human tissue with the investigators. And then the investigators will be sharing the data of their research back to CaRe T1D and also to the broader community through the website that John just showed you. There's going to be reporting results at the annual CaRe T1D meetings, and then developing collaborative projects among the investigators in the consortium. And then the consortium will be planning future activities, what they want to do for collaboration, maybe even tissue, what kind of tissues or the directions we're going to be moving forward. And then eventually we'd like it to really become the scientific think tank, the engine for cardiovascular disease research for type 1 diabetes. So thank you. I think I'm the last presenter. So we're open for questions for all of us. So thank you.
Agree. Thank you. And please go ahead and ask away.
Well, John Yoshika actually had a question here. It's in the chat, but it was directed to me, but I'll just read it out loud if that's okay. Let's say I need to know if levels of gene or protein X are upregulated in type 1 diabetic hearts and kidneys. Such data are available anywhere without getting actual tissues.
So as part of a core lab function, we are not performing gene expression analyses, that would be something that individuals would propose on their own. Would you agree with that statement, Irina?
Yes, we try to characterize, I mean, for best our abilities, we have a staff that are processing cases 24/7, and we do the basic line characterization, including histology, and then certified pathologist, the other PI, like on the project, giving the evaluation that will be available, but so far we do not have a capacity, I mean, more like staff capacity to do the in-depth analysis. And I think that RFA actually designed for finding to do the research on the samples.
Right. So if you want to do 10x or Visium or NanoString or something like that, you know, that would be part of your individual proposal. Sanjoy, I see your hand up.
Yeah, first of all, thank you, NIDDK, NHLBI, Mark and team, and John and team. This is amazing, as Marjorie said at the beginning. And from the perspective of JDRF, two suggestions that I would make. One is to have some kind of an collaboration or discussion with American Heart Association. These projects are extremely valuable, but they do take time to identify targets, to validate targets, to develop therapies, so on and so forth, that will be relevant for type 1 diabetes. To the extent American Heart Association and other guidelines can incorporate some of these findings, that would be helpful. So it's a suggestion. And the second one, on the same lines, is to have some proactive involvement of the industry here. They have excellent therapies coming up for cardiovascular disease, especially with some very recent approvals of novel mechanisms of action that you're all aware of. It'll be really good for them to consider type 1 in their clinical studies, in their clinical trials, because currently they are an exclusion in most of the studies done by pharmaceutical manufacturers. So two suggestions to the team to reach out to AHA and to the drug manufacturers.
So I can hit the first one, and these are great suggestions. Thank you. So Will Cefalu, during his time at American Diabetes Association, was quite active and involved with the American Heart Association in terms of some guidelines for clinical observations. And I have a draft of a document on my computer here that we are composing together with Will, that his goal is to have this article that explains the CaRe T1D movement published simultaneously in diabetes journals plus the American Heart Association journals. So all of this designed to increase the visuals, if you would. Plus, Dr. Yang here is already made contact about trying to present and have a booth at the American Heart Association meeting next year. So these are great suggestions. Yes, thank you. We're on it. So there's a couple questions in here. I'm looking at chat on the time. Again, so time between collection and postmortem. So the average time is about 14 hours from collection to processing. But again, these are all sent in on ice in transport media, like I said, as if they're going for transplant. And we have, again, in the pancreas department alone, we've collected about 650 pancreases. And I'd say out of those 650, there's been less than 5 that came in a condition that was unusable. And we note that, and again, a whole series of QA, QC parameters are used on these tissues. And in theory, we went into this with a lot of suspicions because you have an organ, in that case, that's, largely dedicated to enzyme production. And again, molecular studies, we have over 300 publications using those tissues. So I understand the concern going in, but I think we're doing okay. The suggested order of application, that first question up here in the chat from Aaron Trask. Rahul, do you or Teresa want to address it?
I think it actually Minger might be a little
Minger?
bit better. I mean, I think this is for getting the samples.
Yeah, I sort of answer it in the chat, but I can reiterate. Basically, as Mark mentioned, that since you are dealing with deceased organ donor samples and then you are not getting any PHI from us at all, majority of our experience from MPOD is that this research project are viewed as a non-human research, so it should be much easier for you to obtain the IRB protocol regarding study CaRe T1D samples. And then also we here to answer questions. If your IRB office has any question regarding how the samples are procured, how you're going to get the samples, and how the data is shared, and we're here to help.
Hey, Minger, even though it wasn't expressed here, do you want to just touch on that MTA and IP rights just a little bit to set people as fears, potential fears aside?
Sure. I assuming that NIH just come in the same angles as JDI that we've been working with. In terms of IP rights, the IP belongs to you, the investigators. No, not because you use the samples that we share the IP. No, we want you to have the IP. We want you to generate innovation and benefit from it if you could. In terms of the MTA, it's a very simple MTA. It's just covered between institutes, how the sample is transferred from UF to your institute, how it's going to govern for example, the sample cannot be shared with third party without written notice to us, how the samples is human samples, so with inherent risk handling human samples. So it's a very simple MTA. Hopefully not a lot of negotiation needs to be done. Thank you.
All right, thank you. Alyssa has a question about what happens to the blood samples, and is it possible to get fresh cells? So the way, the part of the goal of today's calls is to hear what the needs of the community are. And this, this was great for us. Again, there's going to be a separate call in December talking about the application process. So right now, I'll let Irina say what happens to the blood samples that come in. And then I'll touch back on. Irina, do you want to share there?
Sure. So the whole blood is processed into PBMCs and plasma, and we also collect serum from these individuals. So this is stored frozen at liquid nitrogen. So I also see questions about where we're taking samples from coronary or peripheral arteries. So the samples are taken currently from coronary arteries from major one RCA, LED, circumflex, and left main. We're working on getting peripheral arteries recovered from the organ donor, but right now we do not have the huge pool. I would say we're working on that.
It's been quite the challenge to obtain that, because again, the families have to consent by which organs they will allow to go, and that's been a challenging one. And plus, you have to have a surgeon that's willing to do the recovery while they're pulling hearts and lungs and intestines and the like. So
Can I add a little bit about special request? Why it was put on that slide is that we have some abilities to do a little bit more than general SOPs. If some researchers really need some, like isolated, for example, cells from myocardium, for example, we can try and pilot this with you, but currently not collecting. So we are open to talk to you, to see your needs. There is a possibility of getting it.
So if you have a specific request, go ahead and email us, and we'll get back an answer for you if it helps in putting your grant together. Rahul or Teresa, do you want to address Peter's question about a VA investigator applying?
Yeah, I saw that. And It's going to be, the MTA is going to be with the University of Florida. So, you know, and we'll try to work it out so it's easy for you to do this, but I can look at the form. NIH isn't going to be requiring anything in this MTA. So I hope that makes it easier for you to get this.
And part of understanding, and Minger, correct me where I'm wrong, is we do need an MTA for this process in part because of requirements put forward by OPOs and then the upstream from them, meaning that we need to, at any given time, be able to give a response to where any tissues that were sent to us, where they're possessed or have been in terms of possession, and what they were used for. I mean, Minger, do you want to?
Right. So that's the something called the chain of custody of the human samples and tissues that we procure from the OPOs. In case the family members request we destroy or return the samples, we need to have this MTA in place so we can go back and ask that a sample that has not been used, please return. So these are part of the unless required process to study human tissues.
Then there was the notion of RBCs versus PBMCs. Again, this would fall into example of what Irina said, something that if there's a request, during the collection process, if this is something you'd want to look at, we're happy to modify or look at and evaluate modifying protocols to do that storage. Let's see, any other questions coming in here? Our goal in setting up this call was to have 30 minutes of presentation, and that worked out perfect, and then have, again, 15 to 30 minutes of Q&A. So we have plenty of time. Rahul, do you, while we're waiting to see if there are any more questions, do you want to give a NHLBI perspective on this or have any comments? Or Patricia?
Hi, this is Rahul Thacker at NHLBI. Nothing salient, nothing timely to add to what Teresa and everyone else have added. I can say from a standpoint for NHLBI and my division in particular, the Division of Cardiovascular Sciences, this is a collaboration with NIDDK and a relationship with the University of Florida group that I think we value and we see the benefit that's come from it. This is an understudied area of research, and it's something that it's on our minds, and we really want to see the most cutting edge research come in and further what we know for mechanistic biology, as well as what we can do down the road for potential therapies, and just with the end goal of being patient care. I mean, none of this will really matter unless we can actually affect a positive benefit for the patients. So we look forward to seeing what sort of applications, what sort of interest this engenders, and any sort of questions along the way. NIDDK, Teresa, and my colleagues at NHLBI and I will be very happy to work with you on. Thank you.
Great. Patricia, do you have anything to add?
I think Rahul stated everything very well. I mean, this is such a unique, singular resource. So we are very much looking forward to seeing the applications that come in to take advantage of it. And as Rahul said, we're looking forward to seeing the science in type 1 diabetes and cardiovascular disease move forward with this. So please send your applications in. Come one, come all.
Yeah. I love that. All right, Tai-Ching, I see you have your hand up.
Yeah, can you hear me?
Thumbs up.
Yes. Okay, great. Thank you for the excellent opportunity. I got two questions. One is regarding getting human tissue from this repository, and is that part of the grant application? That's my first question. The second, you notice that is a collaborative effort. So what does that mean, collaborative? So as a PI, we have to find a scientist in the center, one of the center, like University of Florida or UPenn, or the other. I'm a little bit confused with that.
So let me start with the second question, and I'll second question first, and I'll answer. So no, if we are here to serve those on this call, and then those that may apply that aren't on the call today. Meaning we're a service organization as part of our biorepository. We're here to help promote the notions that Patricia, Rahul, and Teresa said, and the goals Marjorie said. You don't have to list us as collaborators. You don't have to list us as key personnel. You don't, and you don't have to budget for like slides to go. What you need, the key for what you need is a letter from the CaRe T1D saying that your project, or a notion that your project is feasible and then doable, but you don't have to budget for us on anything. Did I overstate that to the CaRe T1D staff, or do you have a different way, a better way of answering that question? Mingder?
Maybe I'll just jump in a little bit, because I think that there's some going to be two parts. So for people who get tissues, and that's everything Mark said, you're getting tissues or you're getting slides, and you're going to use that in your research, then all of that's fine. If you are applying and you become you get a UO1 grant to be part of this consortium, and the UO1 grant is a part of a cooperative agreement, then we would like to have collaboration among that group of investigators and the University of Florida group. And so there it is more of a collaboration, well, like I showed in the last slide, of sharing the data, sharing the results. And we'd like to see, like we've seen in other successful consortium that we've had, for example, on Islet, where investigators work together. And so they have different approaches, and they develop a single project that uses these different approaches, but that's going to take time. So in the beginning, you're going to have your own, project. You get the tissue, you return the data and the results to the group, and then eventually develop a true collaboration with other investigators. I hope that answered your question.
Not really. My question is very specific. So collaborator must be in one of these centers. Is that you are talking about?
Well, Mark, you can go ahead.
Yeah, sure. No, to put in your application, you can put it in as a single investigator if you want. I mean, and then it will go in and be reviewed as a grant. What Teresa is saying is the hope is that the CaRe T1D community will eventually become like a consortium, where at this annual meeting, you'll see data presented. Maybe somebody's going to do single cell analysis, and you'd say, hey, I have this, I want to develop this biomarker, and then this is a thing. And then there's collaborations that would come from that. So that's the notion there. That's how the hope is that this will have an impact down the line. But again, you do not need to list an investigator from UAB or Vanderbilt or UF on your application to have access. Yeah.
Yeah, may I add that I believe the NIH's goal is for investigators to share data back to the consortium. So you take the samples from this consortium you studied and you bring the data back. Once you put the data back, you share the data at the same time, you can also see other investigators' data. So this way would help to move the discovery in a faster pace.
All of that with the notion that you need to publish too. So we fully recognize the need to publish here. So that would not be compromised as part of that. Before we run out of time here, there was a question about how many cases we're going to get. Maybe it went by too quick. The eventual goal is that by October of 24, we have 20 type 1 cases. The website may list 6 now, but I think the number is actually higher. But again, part of the QA/QC here is all of these cases undergo thorough evaluation, medical records, and before we put out a case out there and say this is type 1 diabetes or this is type 2 diabetes, there is a little bit of a lag phase. And so I can tell you that we have more than that. The website, and again, we as soon as we're good to go, John gets them up there. John, do you add anything to that, or Irina?
No, yeah. As soon as these additional cases and moving forward are actually cleared through the Florida group, then they get posted. So that's why the discrepancy between the numbers that you see now on the demonstration that I just showed you and what we expect.
Right. So there's a question on our typical aliquot size, and we've not vacillated, but we've adapted over the years to this in terms of just experience, in terms of not having so much large volume aliquots in every case, so that you exhaust them too quick versus more micro aliquoting. So what is and Irina, do you have anything to that? Then I'll address this review question.
No, this is correct. We can we store at 500 microliters aliquot serum, but we can make it smaller, take smaller aliquot under frozen condition.
Yeah. And then the turnaround time to review has been fast. Usually, at least for our other programs, we try and get answers for back on within 6 to 8 weeks. But now for this program, because it will start out much smaller than our other ones, we might be able to decrease that turnaround time. So this won't be like some programs where you're waiting 6 to 9 months or whatever, like a grant application to hear a score and then funding. We're thinking it'll probably be much more like a 6 to 8 week turnaround.
Let me just add something there. So we're so the samples, there's going to be a limited number of samples that will be given out for investigators to primarily use for feasibility for their methods. So we're not expecting people to have a whole complete preliminary data set on the CaRe T1D samples. There's just not enough time. But if there's questions where you think it would be important to test your particular approach on the collected samples, that's what we would be providing the samples for. So it's not a requirement to get an approval from CaRe T1D to submit an application to NIH. So, and for some people, you may not need to, if you're using blood, then you may not, feel like it's necessary to test the feasibility of the collected samples, and it wouldn't be a negative for review. So, you don't have to, and we hope that it speeds up so people would be able to get these limited number of samples to test the feasibility of their methods out quickly. And so you could include that in your application if you wish. But again, you don't need the CaRe T1D approval to submit an NIH application.
Teresa, Rahul, do you want to, there's this question from Peter asking about, are these going to be similar to R01s, I guess, in terms of size and funding? That'll be in the RFA if it comes out later this week, but do you want to address that?
Yeah, well, I mean, we usually don't say, but they would be, yes, it would be worth your while to apply. They would be, I guess I can say, a generous R01 size.
Okay, good. And then, yeah, so let me go back one thing in terms of feasibility. I'll give you a typical scenario. One of you will have an antibody against compound X using an animal model, and you would really like, you know, a couple slides to show that an antibody in a human setting would work. And so this is what both John and Mingder were speaking of earlier. Contact us. We have a limited number of cases that we're ready to just put the pedal to the metal and get out to you in a process that Mingder can describe, so that if you feel like, although like Teresa said, it's not required, but if you feel like my application will be stronger if I can show at least even a little preliminary data with that, you know, that's what we're here to do. Mingder, would you like to add to that?
I think there's two levels of that. To request the samples for assay optimization or generate some preliminary data, there's actually no application needed. Like I mentioned, just the IRB to cover your research and the a simple MTA. This level 2 is that if you do not get the grant from NIH, the U01 grant, you can still get in samples from us, but in that case, you need to submit an application. The application has to be approved by the committee on the scientific merit. So you may have to find funding from somewhere else, but we welcome investigators come to study these samples.
So we're at the top of the hour. John, I don't know how you want to close this, but again, the last little comment here on the chat is this will be reviewed by a special emphasis panel. So not standard R01. So just on behalf of all the team at CaRe T1D, before John closes, again, thank you all for listening. I hope you learned a little bit more about the mission and the goals of the program on the call. John.
Yeah, I just want to add my thanks too. And as I mentioned before, we will be posting the recording of this webinar plus additional materials on the YouTube channel, which will be made public shortly. And in the meantime, try out the application that is in the care-t1d.org website that you see posted there.
Okay. Well, thank you. And thank you all for listening.
Thank you.
Thank you.